Alright, gang, this is the final section of chapter 16, 16.2D, and if you have not, if you've been working through the reading and if you've been working through the videos and you have not watched the animations that I put online yet, don't even bother with this one yet. Go back, watch the animations in the animation section. They both say chapter 16 on them. And also while you're at it on there, watch the video, the DNA molecular visualizations video. And again, it's a pretty cool thing to see how the actual replication process works in our cells in a little bit more of a molecular view. Anyway, as awesome as the whole process is, what this section is about is what happens when there's mess ups. Okay, and as you read through this section, this last section of the book about errors and how the errors are fixed, it'll tell you that there's going to be an error made when they're putting the nucleotides together. There's going to be an error made once every 100,000 times that they're making a new base pair. So if you're looking at your DNA and we just unzipped some DNA and I look at just one of the strands and it's AA, T, C, C, G, we'll say, and we're going through and we have DNA polymerase putting in the new matching nucleotides, and let's even do this. We'll say this is the 3' end and this is the 5' end of the parent strand. That means that this would be the 5' end here, 5' of our new strand, and so we'd be putting in a T here, and then we would be growing our strand this way. Just some review here because we'd be adding to the 3' end. So another T and an A, and we'll say we put this G in here, and then let's just say that we make a mistake and we put in an A here to go with the C. Okay, that type of mess up happens about once out of every 100,000 times that we're making a new letter. That's how often it happens, but it gets fixed almost all of the time. It gets fixed almost all the time, so that after all of the repair things and the fixing of the mistakes happens, the number of errors that make it through the actual whole process is about 1 out of 10 billion is what it says. Okay. Now those errors, most of the time, they aren't going to be anything that changes a functional part of your DNA. Only about 1% of your DNA is actually used in genes. The rest of it is not. And so most of the time these errors are not a big deal, but every now and then they can cause a problem, and then you get things like cancer, or if it happens to be in a gamete, it can be an error that leads to a genetic disorder. But anyway, the way we can fix these errors, first of all, DNA polymerase, and so I have this up here, DNA polymerase is our first proofreading mechanism, and this would be, the book does a great way of saying this analogy, when you're typing a paper on the computer, and you're typing in T, T, A, G, A, and as soon as you do it, you realize that the A was wrong, you're just going to hit the delete button, get rid of that A, there we go, get rid of the A, and you'll just replace it with the correct G. Okay, and DNA polymerase actually has that ability where if it puts in, again, if we put in the wrong letter here, it'll catch it. The A and the G, their hydrogen bonds don't line up. It's kind of a weird fit, and right away the DNA polymerase will catch it and just get rid of that A and replace it with the correct letter. In this case, is C. All right, so that's one thing that happens. That's the DNA polymerase here. Okay, another thing called mismatch repair. Let's just say that the DNA polymerase didn't catch that, sorry, go back here, didn't catch that C being put in, it was supposed to be a C, but let's say it was an A, and DNA replication gets done. Okay, it's the wrong thing, DNA polymerase didn't catch its own mistake. There is something called mismatch repair, and the G and the A here, as they're not matching up, there's actually enzymes that would go along this and basically scan for mismatches. This would kind of be like your editor, somebody who is going to read if you're typing up a large report or a paper, excuse me, or a book, and an editor comes along and you've done all of the, you've caught everything that you can catch as you're reading through all the mistakes, and an editor will look at it and they'll get to this and they'll say, oh, well, this is obviously a mistake, and they'll fix it for you. Okay, so they get rid of that A. See if I can just get rid of the A here. That's going to get rid of that. It'll get rid of that A and replace it with the correct thing, the C. Okay, and there's a series of enzymes. The book doesn't get into much detail on that, but basically they scan up and down the DNA and look for mismatches, and as they catch them, cut the bad nucleotide out and replace it with the correct one. What is detailed in the book is a different type here called, let me delete all these things, called nucleotide excision repair. Okay, and this is one that you're going to need to know. It's a whole process, and this one deals more with when we have a mistake in the DNA, actually like a problem that has formed in the DNA. So the example that they use is a mutation that has happened because of ultraviolet light. And what we have here is a segment of DNA that we're showing right here, and that segment of DNA, you can see this little buckle in it, and there's a problem there. That buckle is what they call a thymine dimer, and what a thymine dimer is you've got the sequence of DNA, it's C, G, C. If there's two T's beside each other and another G, if you hit this, zap this thing with ultraviolet light will actually, and there's supposed to be a C, G, C, G, A, and there's hydrogen bonds here between the thymines and the adenines, okay, holding them together. If you hit this with ultraviolet light, let's say this is a skin cell, hit it with ultraviolet light, what will happen is these bonds between the T's and the A's will end up being removed. Let's see if I can get rid of them. Okay, and they will be replaced with a bond between the thymines. They call this a thymine dimer. And so the thymines that are right beside each other will end up bonding to each other. It's the high energy from the ultraviolet light that'll do that. And this can cause a problem in the future where these two T's that are stuck together, that in the next round of replication, you won't be able to match up any A's there, and it ends up causing a problem. That's why ultraviolet light can lead to cancer, because it increases the chances of there being a mutation. So for this one to be fixed, this is going to be found your body actually has a series of molecules, enzymes that are going to come through here and basically recognize this buckle in the DNA and say, hey, there's a problem there. And so something called a nuclease, and that's a word that you'd want to know, a nuclease is an enzyme that's going to go through and first of all chop out the problem area. And so you can see it here, it's chopping right here, and actually remove this segment of DNA. Once that segment is removed, then a DNA polymerase can come along. There'd be an available 3' end, and the DNA polymerase can come along and fix the spot, fill in the correct nucleotides. Once it has filled that segment, then DNA ligase is going to come along and do what it does and just connect it all together again. Okay, that's DNA excision repair. And that is how your body can fix things. Like you can be exposed to ultraviolet light and not be guaranteed mutations, because your body will fix this stuff. Okay. People who are highly prone to skin cancer, they may have an error in this whole mechanism that's supposed to fix these thymine dimers. Okay, people that are, yeah, and we'll just kind of stop it at that. But that's excision repair. That's the fixing of errors. Okay, the next slide here, we're going to look at another thing that happens through replication that has to be fixed that can that could ultimately lead to some problems, and we'll get to the next slide. Alright, this next problem, I'll try to go through this quickly, is the problem of chromosomes getting shorter and shorter every time that we replicate DNA. And if I take a look at this is supposed to be the end of a chromosome, and if you can imagine that our origin of replication was back here somewhere, again the blue would have been the original DNA that was connected together. So we've unzipped it. We've had DNA polymerase, and again back here would have been the origin somewhere, so there'd be a primer there that would have had to have been replaced. And then the leading strand, as we go this direction, would have basically run to the end of the chromosome and replicated everything. Not a big deal. The lagging strand, however, had to lay a primer, work backward, lay a primer, work backward. And all of these primers could be removed and replaced with DNA. If you remember, DNA polymerase 1 does that. And then ligase fills in the gap. Until we get to the very, very end of the chromosome. A primer can be laid there, primase can do that, and then DNA polymerase 3 can build one from that primer. But if we zoom in at what happens next, there is a problem that you will see as we go down through. If we zoom in on this, this last fragment here, we get, here's our primer, here's a Okazaki fragment, here's the other primer. We're going to replace that primer with by using DNA polymerase 1. But in the end, when we remove this primer that's all the way at the very, very tip, there's no way for DNA polymerase 1 to build this section of the DNA. The reason it can't is because, remember, it has to build onto a 3' end, but this is the 5' end of everything, right? Here's the 5' end. So DNA polymerase 1 is not able to fill in this gap. So basically, this side of this chromosome is going to be short. It's going to be left short. If we were to go through the next round of replication, which is, as I expose this here, delete this. If I go through the next round of replication, where we unzip these two, because this will be in the next cell, unzip these two, the bottom is no problem, right? The bottom is going to be fine. But the top is now going to be shorter, and we have a shortened chromosome, okay? So this actually happens in your cells. So the cells in your body now are their chromosomes are slightly shorter than the ones when you were just an embryo, okay, because you've been cutting off just a few nucleotides every single time that a cell replicates. Not a huge deal for you. There is something called a telomere. Okay, and I should write that on here, a telomere. And what a telomere is basically all of your chromosomes are designed with a very large section of DNA on the end of them that is useless as far as coding for any kind of genes. And so you've got a very large segment of DNA that is, how do I say that? That you can eat through before you get to any actual genes that could cause a problem for you. Okay, and that is enough for you to last your entire lifetime. That's how long those telomeres are. Okay, and so that's not a huge deal. Where there is a huge deal is if we look at this process, if it were happening in a cell that was going to become a gamete. So let's just say, and they call these germ cells, or cells that are like the original cells that make new cells. So if these were, if this chromosome right here that we were looking at, this DNA, if that were in a germ cell that we're going to make an egg, okay, that means, so this germ cell is going to split and it becomes, there's a whole bunch of cell divisions that happen before you finally get to an egg. So you have mitosis happening 3 times, I think, and then meiosis finally happens. But anyway, let's say that this ended up being the DNA that ended up in an egg. That would mean that your son or daughter, their chromosome is slightly smaller than yours. And your chromosome would be slightly smaller than your parents'. And your parents' chromosomes would be slightly smaller than their parents'. And if you were to compare people now back to the very first people, then the chromosomes would be significantly shorter. Okay? Well, ultimately that could lead to the extinction, excuse me, of our species. If our DNA is getting shorter and shorter, eventually we would die off. We'd get to a valuable gene, and people wouldn't be able to live anymore. And this would happen to every single organism out there, that they would just not be able to live anymore. So, a really, really cool thing, at least in my book, that exists is in these germ cells that lead to making gametes, there is something that will rebuild a telomere. Okay? It's called telomerase. Let me write this. Or telomerase. Telomerase. And basically what it does is it has the ability to build on to the end here. And so on this this segment right here that was going to become the egg or the sperm or whatever, we can just build on some more telomere. Again, it's wasted DNA, so it doesn't matter. I think they usually are just a repeat of certain sequences that mean nothing. You can just build that on there, and it gets rid of this ability to, or this threat of chromosomes getting small enough in the future to cause problems. Anyway, telomerase or telomerase. Anyway, something I'd love for you to do is think through like the evolutionary principles behind that, and just open-mindedly or critically think through how would something like telomerase be able to make like a preventative measure that seemingly doesn't have much of an advantage until generations from now. So there's this preventative measure, and I don't know if you're kind of tracking with me there, but think through as far as evolution of new molecules and things like that. What would the benefit of having telomerase be for organisms now, when their chromosomes are already large, kind of thing, for them to specifically have that in their germ cells. Anyway, kind of think through that. I think that's all the time we have for this now, or at least all the time I'm going to take. That's the end of the chapter. Hopefully, if you have questions, you're asking either in class or on the website. And yeah, we'll see you in class.